Good morning, everybody. Thanks for those who are currently logged in. We are about half our attendees logged in that we're expecting, so we're just going to wait for a few more minutes. And at about 2 or 3 minutes after the hour, we will start the presentation. So just stand by, please. Okay, good morning, everybody. Looks like there's a few more on here now, so I'm going to go ahead and start. Welcome everyone to this installment of the IPHC 2022 Seminar Series. Today it is our pleasure to have Dr. Noëlle Yochum presenting conservation engineering approaches for supporting fisheries sustainability in Alaska. Noëlle leads the conservation engineering group from the Midwater Assessment and Conservation Engineering Program at the Alaska Fisheries Science Center in Seattle, Washington, conducting collaborative research with the fishing industry and other partners to evaluate fishing gear performance. This is done through innovative research on fish behavior and biology and gear design in support of fishery sustainability. Projects include evaluating, quantifying, and mitigating bycatch, discard mortality, and other fishing-related impacts, and developing and applying new technology to observe fishing gear and fish behavior in situ. Noëlle has 2 decades of experience working with a variety of fisheries and fishing gear types in the US and abroad. The IPHC has been fortunate to collaborate with Noëlle on several projects through NOAA's bycatch reduction and engineering program, including current projects pertaining to reducing Pacific halibut bycatch in trawl nets through the use of LED lights in collaboration with Dr. Lamelli from the Pacific States Marine Fish Commission, as well as on protecting catch from marine mammal depredation. Noëlle has a master's degree in marine science from the College of William and Mary at VIMS and a PhD degree in fisheries science from Oregon State University. She's also an affiliate faculty at the Alaska Pacific University. Questions during today's presentations can be submitted via the Questions tab on your control panel, and these will be read aloud by the moderator at the end of the presentation. This seminar presentation will be recorded and should be available on our website later this week. And with that, Noëlle, you're welcome to activate your audio and start your presentation.
Thank you so much, Claude. Can you hear me all right?
Yes, you bet.
Wonderful. Thank you so much, and good afternoon, everyone, and thank you for coming to hear my presentation today. With that wonderful introduction by Claude, he's been able to give you a little bit of a background about who I am and what I'm doing. So just really, again, in brief, we're leading the Conservation Engineering Group working at the Alaska Fisheries Science Center with my colleagues David Bryan and Catherine Wilson, and we have contractors from Pacific States Marine Fisheries Commission, and we work with undergraduate and graduate students as well. And Claude gave a nice overview about what we do as a group. And so some of the categories of the work that the research projects that we take on are related to gear selectivity, thinking about reducing bycatch, also thinking about how we can quantify and mitigate bycatch mortality, thinking about animals that interact with the gear or are discarded. Also thinking about how fishing gear interacts with habitat, derelict gear as well, so the lost gear, and thinking about how to modify gear in order to make it more energy efficient. So essentially anything coming back to gear performance and how to make fishing more sustainable falls into this wheelhouse of engineering approaches. I want to highlight one aspect of this is really the collaboration is really key to everything that we do, and you'll find throughout this highlighted the importance of working with the fishing industry and other science partners that really makes this work possible. Because I promised in my abstract to give some idea about conservation engineering and what that field of study is, I was really excited for the opportunity to share with folks what that's all about. It's something I care a lot about, and so I wanted to provide some context and give an idea of some of the projects we're working on, and then I'll focus more on one particular project to give a deeper dive into how we do conservation engineering work. And Claude already mentioned this. So Claude and Ian Stewart are doing a project at the Halibut Commission looking at protecting protecting catch from whale depredation, and thinking about gear modification for that. And we are very happy collaborators on that project. Also, I'm working with Rebecca Hain, who's at the Alaska Fisheries Science Center, and as part of her master's work, she's doing work thinking about fish vision and how fish perceive fishing gear. Again, another project we're collaborating on. And a lot of the work that we do really falls under this umbrella of thinking how we can modify fishing gear in order to reduce the capture of unintended animals or bycatch. And so I wanted to give a few examples of the projects we're working on. One is the project that Claude mentioned. Derek Jackson is a master's student at the Virginia Institute of Marine Science, working under Dave Rudders, and in collaboration with Mark Lomelli and Claude and others at the Halibut Commission, we are looking at how to reduce Pacific halibut bycatch in the bottom trawl fisheries using artificial lights. We're also working on a project to try to modify the pot for snow crab to select against small males in order to reduce discard. And then again, the project I'll be talking about in more depth today is thinking about how to reduce Pacific salmon bycatch in the Pollock fisheries in Alaska. So that last category of research projects that we're working on fall under this umbrella of bycatch reduction devices, or BRDs, which is simply a way to modify your fishing gear in order to reduce the capture of unintended animals or bycatch. So basically making your gear more selective. The classic example of a BRD is the turtle excluder device, that first picture you see on my screen. And that is basically a grate that allows shrimp to go through, but then forces mechanically those turtles to go up and gives them an opportunity to escape. This turtle excluder device, or TED, is a really classic example of a BRD and has been very effective. And then just to show the range of these types of BRDs, here's one in the middle. This is from HookPod. This is a way to protect the hook as it descends for long line gear in order to reduce seabird bycatch. And then the last one is an acrylic bead that has been placed on a gill net. And this is work that's been done out of Germany. And what they're doing here is allowing that gill net to be more visible to mammals. So when they echolocate, these acrylic beads allow that net to be more visible to them. So it's like creating an acoustic wall for them to detect that and minimize entanglement. So these are, just to show you the spectrum of some of the different ways that you can modify your gear, these BRDs, in order to make your gear more selective and reduce the capture of these animals that you don't want to harvest. So regardless the gear type, regardless of what you're trying to accomplish, really when you're developing a BRD, it comes down to thinking about the animal you want, your target animal, and then thinking about the animal you don't want, the bycatch animal. And key to developing any BRD is to really think about how are they different so that you can try to select against one and except for the other. And you can do this through thinking about morphology. So with the turtle excluder device, or the TED, it's clear that shrimp and the turtles have a very different morphology. Their body shapes are different. Obviously physiology too, they have different swimming abilities. So the TED relies on those differences in order to create that BRD for that system. For animals that have similar morphology, maybe again with the physiology, maybe different swimming styles. Also, we can look about the sensory systems and how those might be different. So maybe different visual systems, different responses on the lateral lines, different responses to water flow, or maybe behavior. So some animals, when they're startled, they might automatically swim down, but others, maybe they swim up. And so really thinking about how these animals are different is really key into developing these BRDs, because you want to maintain your catch and get rid of the other. And so this is sort of the fun puzzle that is developing a BRD in its sort of distilled form. So today, and that's just some background on conservation engineering, I'm hoping today to really dive deep into one fishery and one BRD project that we've been working on for the last few years, to give a more concrete example about how conservation engineering is used in order to make fisheries more sustainable. And I'm really excited to tell you about the work we've been doing here. And so this is the Alaska Walleye Pollock fishery. And obviously they're targeting these pollock, but also they're trying to contend with the issue of Pacific salmon bycatch. So Chinook and chum salmon are primarily caught as bycatch, and these are managed as prohibited species catch. And for Chinook, the capture of Chinook can lead to closure of the fishery. So there's a lot of incentive to reduce bycatch of the salmon, in addition to wanting to make the fishery, to minimize bycatch and to accommodate target fisheries and the health of the population. So there's, in trying to come up with a way to develop a bycatch reduction device to reduce salmon while maintaining the capture of pollock, you can see that this is a little bit more of a challenge than a turtle and a shrimp, in that they're very similar body shapes and they're found in similar areas, relatively similar swimming styles. And so trying to think about how they're different in order to come up with a BRD solution. And so back in the early 2000s, it was the thinking of, well, the big difference between these animals is that, well, salmon are stronger swimmers. So we know that salmon are anadromous and have that stage of their life where they're swimming upriver and have to be really powerful swimmers. And so starting in 2002, really driven by the fishing industry, was trying to come up with ways to develop a salmon excluder, a type of a BRD, in order to allow salmon to escape once they're in the pelagic trawl used to harvest pollock, in order to allow them to escape, and then trying to think about how you can design that and taking advantage of their swimming abilities. And so really the idea behind it is basically creating an opportunity for the salmon to get out of the net after they go through the net, and then end up back sort of towards the end of the net, as you can see there in that last taper section, before they get into the cod end or the bag that's retaining them. So there's been a number of designs of salmon excluders since 2002. John Groover, John Govan, Mike Stone, others that came before me working on this project, and coming up with different designs and trying to think about how best to encourage escapement of salmon. And so while there's different designs, really the core idea behind all of them was that a pollock will swim back and past whatever escapement portal or open area is provided, whereas salmon have the opportunity, because of their strong swimming abilities, to go up and out or down to the side in whatever location that opening is in order to escape. I'm not going to go through all the iterations of the development of excluders over the years, but I will share a couple of designs that are more commonly used today. So this is called the flapper design, and it's just a ramp that goes down in the net. It's actually typically placed in the intermediate, which is between the net and the cod end, and that allows the pollock to go back and past that area into the cod end, whereas the salmon would be able to swim up and out that opening. There's modifications of the flapper where there's actually an opening right in the ramp as well. This is a much better diagram developed from the Govan and King 2021 report. And you can see this is a side view, and you can see the fish would swim back, and down below that blue area, which would be the ramp, and then the salmon would swim up and out as an opportunity to escape. Another commonly used salmon excluder is this over-under design, and that's basically the same idea as the flapper, but it has two ramps. I guess one's more of a ramp and it's weighted, the other has flotation, and it allows salmon that are caught to escape either up or down. And again, a much better picture from Gavin and King 2021. And again, you can see the fish would swim through those two blue ramps and go back, whereas the salmon would then go through that and then either turn around and go up or turn around and go down. And up in the top there, you can see a model of that in the fin tank. So despite the success of these excluders in resulting in escapement, which they have, there is high variability in those rates. And so this is a figure I put together where your y-axis there is salmon escapement rate. And you can see there's quite a bit of variability. So in the light gray is reported estimates of salmon escapement in the Bering Sea from different trials. The Gulf of Alaska is in darker gray. You can see the funnel, flapper, over under different excluder designs, and then thinking about Chinook and chum salmon in these different seasons. And really the takeaway here is there really aren't a lot of patterns in understanding why salmon escapement is so variable. Can go from anywhere to 50% to 0% in a given trial. And so while these are clearly allowing for escapement, there's still a lot of variability in those rates. So the goal really is to think about how to make those rates more reliable so that we know that we have a tool that we can depend on even more than we do now, and then to try to see if we can get those escapement rates even higher. And the goal in developing any BRD, bycatch reduction device, like these salmon excluders, is not only to have high escapement rates, but also to make sure that whatever escapement opportunity you're providing is not leading to increased pollock escapement. As you might imagine, if you have a lot of pollock escapement and you have to go and conduct many more trips, you could end up with more salmon bycatch in the long run, which sort of negates the benefits of these excluders. And so in trying to take that on in the conservation engineering group, we wanted to take a more mechanistic look and to see how our excluders actually working and how are salmon behaving in and around them, to think about how to improve these designs and really understand what are the limiting factors in getting higher escapement rates and what's causing so much variability. So regardless the design, whether it's a flapper or an over-under, any design, really the key elements in designing these excluders is access, meaning that the salmon need to have access to some opening. They need to be able to perceive that opening. They need to know that there is an opportunity to escape, and they also need to be motivated to escape. They need to actually actively want to take advantage of that, or they need to mechanically be forced to take advantage of that opportunity. So regardless, the design, these are really the key elements to an excluder. And so thinking about that within the conservation engineering group and with a group of wonderful collaborators, we developed the rope tube and funnel, or the RT&F excluder, partially to test out this design concept, but more importantly, really to think about what would happen if we had an excluder with nearly full access to escapement, and just give them as much opportunity to escape as possible, and then see what they do with that, to really think about these key elements I mentioned. And so what you can see here, so this is a drawing of a pelagic trawl. And as the pelagic trawl goes back, that last taper section is where the net is ending. And then you have the caught end where the catch is retained, and that intermediate netting between. And so this was placed, was intended to be placed in that last taper section as the trawl net is reducing its circumference. And the idea is to have a funnel that goes through that mesh, and that has ropes surrounding it that go back and rejoin it with the rest of the trawl. And the idea here again is to have the fish go through the funnel and emerge in a completely open area. Granted, they're surrounded by ropes, but those are sort of needed to maintain the integrity of the trawl. But this was the way we thought we could get at the most open area. And so thinking back about those key elements, again, for access, we wanted to provide this 360-degree open area. Not only is it fully open, but it also doesn't require the salmon to swim up and back. It just simply provides an opening. And then in regard to perception, the design of this helps to break up the visual pattern. So as you might know with salmon, they're going to go down and they're going to see trawl, trawl, trawl. It's all looking very much the same. But having that opening, it sort of breaks up that visual pattern, as well as some of the patterning we have on the excluder itself to sort of make them notice that they're in a new place. We also wanted to use water flow, more importantly, as a way to help them perceive that area, which I'll talk about in a minute. Similar to increase the motivation to escape, we wanted to use water flow, and I'll talk about that here. So in designing this excluder, I worked really closely with a fluid dynamics scientist in Germany, and he partnered very closely with us on the project. So we provided the design and we went back and forth, and he helped us think about the mesh size, solidity, ratio, these various components that really affect water flow and how netting is constructed. And so, or the excluder is constructed using these netting. And so he modeled this with the computational fluid dynamics modeling to really think about how water is going to flow through this. And so on here in the diagram below, you see the red is faster moving water and the cooler colors is slower moving water. And we were trying to achieve what you see here, which is to have faster moving water going through the funnel. So there's no congestion, no clogging, that they're going to go through that. And the pollock will go through quickly, so we reduce our loss of pollock, while at the same time trying to generate areas of lower flow in and around that area to help them also feel that something else is going on there. So salmon are very sensitive to water flow. They can detect changes in water flow. They're very finite. So giving them something to feel and feel that they're different, but then also in regard to motivation, knowing that salmon like to go into those lower flow areas, hoping that it would draw them over to those low flow areas, which would then put them right at the escapement. So following the design process, working really closely with Dr. Bredeman, the fluid dynamics specialist, we put together a model to test in the flume tank. So this is in Newfoundland. This is a flume tank, which is a massive tank that also has the ability to generate water flow so that you can put model fishing gear in it in order to evaluate it and test it before taking it into the field. So that's us working at this flume tank, and there's a model of the image you saw before, and us as a group working and modifying it as we go. And one of the other advantages of working at the flume tank is we were able to collect flow data to see if our predicted water flow values were correct based on Dr. Bredeman's models. So you can see here in this circled area, this is a water flow measure. And based on that, we were able to look at the water flow through this device and confirm that we were getting the water flow that we were hoping for, which is, again, faster water flow moving through the funnel and then having areas of lower flow in and around those open areas. And again, this is in more detail in Bredeman et al. In 2019, for if you're interested in reading more. So following the work in the flume tank, I was able to finalize the design, and then I worked with Swan Nets to make an actual full-scale model of this. And that was tested in the field in 2019 and 2020. And in this video, you can see the end of the funnel. So the funnel is what you're looking at there, and the fish are coming out of it and going back through into the trawl. And that open area that you can see the ropes, but that is all open area for them to escape. So this is essentially how it looks, and you can see that flow does move pretty well through the funnel. So the results of the 2019 fieldwork were published in Fisheries Research. So if you're interested in more details about that, or if you're interested more about the development of the excluder, you can read it in this paper. And this also gives me an opportunity to acknowledge my co-authors and my colleagues who worked on this project with me. And then I won't make you go read it to give you the takeaways. I'll provide a little bit of information about the results from the study. and talk about that in relation to access, perception, and motivation. So with regard to access, on average, we had 58% salmon escapement with this RT&F design, which is high escapement, which is great. But I think the bigger takeaway is that there was 42% retention. So despite there being 360-degree fully open area, fully access to escapement, there was still quite a bit of retention of salmon. So what this told me was that the location and design of the escapement area, where that opening, where that hole is really not the limiting factor. So thinking about, is it on the top or on the bottom, that's important, but giving them full access really didn't fully address that. So access is not the limiting factor in trying to develop these excluders. In regard to perception, what I noticed and what you could probably see is the fish go through that area very quickly. Perhaps they're going past there too quickly. And so as a way to think about how to fix that is maybe making that area more perceptible. And then thinking about how to do that, perhaps using artificial light as a tool. In regard to motivation, really what was surprising to me is they really had, I guess this is sort of anthropomorphized, but they did not seem to have much interest in escapement. So they weren't really looking for opportunities to escape. So much so that you would see some salmon go down that area and back down into the trawl and turn around and go back through that escapement area again and simply go right back up into the net. So there was really not a lot of motivation to escape at that time. So I'm thinking about how to address this. One of the things that was really interesting is in 2019, in reviewing the footage, what you can see is that a lot of the escapement is happening at haulback. And at the time of haulback, you see a lot of the salmon coming back from the cod end area and swimming forward and escaping during that time period. And so that was really interesting to me in trying to think about, well, what is triggering that forward movement, and can we take advantage of that to learn about how we can better design these excluders to try to get better escapements right there at the excluder. So here's a video of that. This is from the 2019 video footage. You're looking at the end of the funnel, and you can see salmon coming forward during haulback and taking advantage of the open area again and escaping. But again, these all had gone past the excluder at one point and are only escaping now during haulback. So in order to try to learn more about this, David Bryan, who's in the conservation engineering group, has taken on this project of trying to understand salmon behavior to really understand what are the triggers for that forward movement so that we could learn maybe strategies for improving salmon escapement. So he's looking at the phase of the fishing process, so looking at the fish behavior during setting, or not so much setting, but fishing and haulback, and thinking about things like ambient light, tow speed, depth, other variables that are triggering this forward movement. And so in order to accomplish this, we went out and put the RT&F excluder in the same place it was in 2019 and put these ruggedized trawl cameras at the excluder and then along the way towards the cod end in order to capture video footage of salmon and the way they're behaving, and linking that with some of the other data we were collecting around light and depth, water flow, temperature, etc. And so this is still an ongoing analysis. David has not completed this yet, but he did provide some preliminary data for me to share with you. And this includes some work or some analysis where the camera at the cod end, so this is the camera looking right into the cod end. David here has plotted, this is for 2 tows. So your x-axis is the duration of the tow, and the y-axis is your depth. So you can really see the gears descending, fishing, and then that solid vertical line at the end there is when haulback is starting. And that dashed vertical line is when the doors are up on deck. And the yellow dots are when David saw a salmon moving aft, so going into the cod end area, and then the blue dot is when it's swimming forward. And what's really interesting is you can see there's forward movement during the whole of the tow. But what is kind of exciting and interesting as a preliminary look is that when the doors are on deck, you have a disproportionately higher rate of forward movement. In 2019, you can see there's an especially high forward movement as it gets into shallower depths. So not making any conclusions based on this. This is an ongoing analysis, but it's exciting to think about what we might learn with these triggers that might help us develop better BRDs. And so then in addition to addressing motivation, there's also the potential to try to think about how we might deter passage past the excluder, so getting them to stop after they've gone through and not go any further, or perhaps trying to attract them to an open area of the net, so trying to get them to come to that open area. So again, trying to use maybe artificial light for that. So I'm going to focus the rest of this talk on the work that I've done or we've done around artificial light and trying to affect perception and motivation. And again, this would be artificial light to augment the BRD or augment the use of a salmon excluder. In regard to perception, trying to use light to make the escapement area more perceptible, given that they go by it, that open area so quickly, which could mean illuminating the area or just breaking up that visual pattern. And then again, in regard to motivation, really trying to deter passage into the intermediate, so getting them to stop after going past the excluder. And that would only work if salmon exhibit a negative phototactic response, meaning that they move away from the light source. Or the other option would be to attract them using light to an opening, and that would require the salmon to have a positive phototactic response, meaning that they tend to move toward a light source. So thinking about sort of some anecdotal information to inform the study, here is a video provided to me by John Govan in a study he did, and what you can see here is a salmon going up that flapper ramp, and it seems that he's running straight into that camera light. So there's a camera at the top of the ramp. And there are those that have used cameras in their fishing gear and have thought and seen this behavior and thought, well, perhaps it is a positive phototactic response for salmon. They are actually, quote unquote, attracted or moving towards the light source. However, again, in this video provided by Govan, that this is now from the camera that you saw before looking down that ramp. And you can see the salmon come up, bump into it, and escape. However, this different perspective really lets you see that, you know, there is potential that it's not so much that they're attracted to this light, perhaps that the camera is simply on its way out, or perhaps the camera is generating eddies or areas of different water flow that it's being attracted to being attracted by the salmon. Because the camera that you see in the back, there's a glowing light that's further aft, and we did not see that same behavior for the cameras that weren't right on their path out. So some different things to disentangle here. To add some more information, Dr. Mark Lamelli at Pacific States, who works on the West Coast, is working in the hake fishery, so different fishery, different gear, but this is also for salmon excluders, and he did find success in adding light to his escapement area in that fishery. I don't believe that the fishermen are using those regularly, but he did have some interesting research results. So lots of pieces to think about. So taking all of that information, I'm going to talk a little bit about 3 studies that we did to try to address this question about artificial light. The first of which is thinking about, are salmon actually, do they have positive phototaxis, and would they just move towards a white light like we saw in that video? And then also trying to think about the different properties of light and how that might bear on behavior. And then the third one is a study that we did looking at how we might increase rates using blue light. So I'll start with this first one, which is really a very simple question, which is, are salmon quote unquote attracted to light? If we put a light into a net, could we just draw them there? And of course, that would make our lives very much easier, I think. In order to answer this question, I went out in a pelagic trawl that is used for pollock, and in the area where you might expect to insert an excluder, we took the excluder out and simply added these timed lights. So there'd be one side of port, the port side would be lit, and then it would go starboard and back and forth for a 15-minute duration, and really just trying to see when the side was lit up, are salmon swimming towards it. Because as you might imagine, trying to use white camera lights is going to throw off a study trying to look at behavior. We wanted to think about a workaround. And so in order to minimize the impact to behavior, we used cameras illuminated by infrared light, which were put together by Scott McIntyre, who was formerly in the group. And so the results of this are still preliminary, but the things we looked at was these different behaviors. So thinking about passive versus active swimming, the direction that they were moving, whether they were moving toward a cod end or the mouth, between the panels, the starboard and port panels, left and right panels, and then thinking about their orientation and just trying to understand it. How are they responding to this light? And what's interesting is 63% so far, and the salmon assessed were moving passively. 37% were active, with a total of 55%. So the majority simply moving passively, facing the mouth of the trawl. And what that looks like is this in this video. And again, this is still not fully analyzed, but this is most of what we would see is the salmon just going past that light. So not really exhibiting a clear or consistent phototactic behavior when moving aft in the trawl. So it was not a clear, oh, they're moving towards the light, like we were hoping based on that video we saw. And then also from this, really learning that infrared light is challenging and it really has a reduced field of view. So thinking about going forward with these projects and trying to think about how we can better observe behavior with the cameras, and thinking about, well, is there a different color of light that we can use to illuminate our camera's field of view in order to see further? That infrared was not enough. And in order to do that, we really needed more information about salmon vision, because all at the time that's known about salmon vision is when they're in their freshwater residency. And just given the ontogenetic changes that they have, given their anadromous life cycle, we were worried that there would be real meaningful changes in their marine residence. So, not being a fish vision expert, I partnered with Dr. Laubrit, Rebecca Hain, and Dr. Ellis Lowe to go out and collect information about Chinook vision in their marine residence. And from their work, came to the conclusion that I would use red lights in the far red, so a lower wavelength than infrared, which gave me better field of view for the cameras. And then that led to the next study, which was thinking about light properties and how different light properties might differentially affect behavior. As you might imagine, going out into the field and testing lots of different light properties would be very time consuming, and I'd be concerned that it'd be hard to get sufficient sample size to really tease that apart. And so at this time, I wanted to do a laboratory study to look at light intensity. So thinking about really bright white light versus dim white light. Also thinking about wavelength, or the color of the light, and how the different colors might affect behavior. So thinking about blue and green and white and red. And then I also looked at strobe rates. So 1 second on, 1 second off, 0.25 seconds off, 0.5 seconds on, 0.25, 0.25. So 3 different strobe rates in order to see really how are the salmon responding, and did the light properties make a difference? And then trying to use this laboratory study to inform behavior in order to know how to proceed in the following field studies. And again, the question being, how is salmon behavior affected by light properties of intensity, wavelength, or color, and strobe rate? So to address that, I worked really closely with Dr. Barajikian and colleagues at the Northwest Fisheries Science Center, who have this fantastic laboratory set up. So in these tanks, I was able to get a company to manufacture these blackout, basically tents that went over the tanks. And inside the tanks, the arrows are pointing at lights. And you can see the lights would illuminate one at a time. And I would observe the reactions of the salmon using cameras, and then using a behavior tracking software to see how the salmon moved in response to the light, both when lights were illuminated and during control periods. And so this paper is published in CJFAS if folks really want to know the details, and also gives me a chance to acknowledge my wonderful collaborators on this project. But without going into too deep a dive, I'll give some basic results, which is results suggest a negative phototactic response rather than positive. And so a common observation was when the light went on, you would see the fish swim away, and they would tend not to go back into that lit quadrant for the duration of the light exposure. So this is the fish tracking, and you can see the arrows, this is where the light is, and you can see that the fish tend not to be swimming in those areas. So really more of a negative phototactic response, or moving away from the light. And then the effects of the color and strobe and these different properties really did differentially affect the behavior. So some general results. Here you can see standardized distance of from the lights. So this is relative to a control. So if it was at 0, that's indicative of similar distance from the light to the control, whereas if it's greater, it's showing negative phototaxis, meaning that it's further from the lights than a control. And below the line is positive phototaxis, meaning it got closer to the lights. And you see here we've got the non-strobing and 3 different strobe rates for the different colors. And so what's interesting is it seems to have even further distance from the light for the strobe lights. And within non-strobing, the blue had the least extreme response to the lights. And then again, looking here, this is again standardized. This is the non-strobing and the 3 strobe rates for the different colors, but this is over the duration of exposure to light. And again, above the 0 is negative phototaxis or moving away, and positive phototaxis below, meaning getting closer to the light. And just taking a look at the non-strobing, what's interesting is you can see that for some of the colors, there is a bit of what you might call habituation. So they get a little bit closer and closer to the light over time, which is more the case for the red and the green. Blue, again, there's a lot more close proximity to the light than the other colors. But for white, it might there's a bit of a negative slope, but really they don't ever get quote unquote used to that light, the presence of that light. There's also an effect on swimming speed. So for strobing and non-strobing treatments, again, on the y-axis here, we have swimming speed, and the dashed line that you can see is that's the control speeds. And what's interesting is for non-strobing treatments, there seems to be a bit of a startle response at first, and then they actually increase their swimming speeds. Whereas with the strobing treatments, you don't see that same response. And to show you that here, this is the combined swimming speed again, the white being the baseline or the control, and then you can see the different colors for the strobing and non-strobing. And again, you can see a faster swimming for the black, which is the non-strobing. And what's really interesting is for the 3 different strobe rates, there's a similar pattern among them showing that the strobing rate does seem to have an effect itself. So again, as a quick summary, really how salmon behave in response to light really does depend on the light properties. And so it's really important to include those in thinking about how you might use light to affect behavior in regard to bycatch mitigation. But I will say that you cannot extrapolate directly to the trawl setting. So as you might imagine, how a salmon is going to behave inside the lab study is going to be different from the trawl. In the trawl, there's many more stimuli. They're experiencing noises and vibrations, and fish are all around them. So it's really, you can't necessarily take what you learn from the lab to extrapolate that and assume that you're going to get that same response. But what it does do is it allows me to help select properties to use for future studies. And so in thinking about perception and trying to make that area more perceptible, it seems that blue is probably the best given the responses from the lab. And thinking about trying to deter passage and having them not go any further, that white strobing light seems to be the best thing to try based on those laboratory results. So that leads to the third and final study that I was going to talk about today, which is thinking about how to use blue lights to increase perception of that open area. And so in this RT&F excluder, adding blue lights around the roped area. And this is what it looks like in real life. So this is blue lights placed around to try to make that area more perceptible. And then using these trawl cameras illuminated with those red lights that I previously mentioned. So right now we're currently generating the results for that. So I don't have any results to present with you today, but the idea is to compare escapement rates for blue lights versus no blue lights. And then also to think about salmon behavior and see if the blue lights affect where they escape or how they're engaging in that open area. And then also thinking about, are they escaping more actively or passively with those lights? And those first 2 questions were part of Michelle DeRoy's undergraduate senior project. So she was an intern with us and did a preliminary look at some of that, but not with the blue light. She just looked at no blue light and had some interesting results where without the blue lights, the majority of the animals that she saw escaping were escaping from the top. And then the green you can see there, those are when we couldn't determine whether or not they escaped just because of the camera angle. And so there's more to be done there, and we're going to continue that research and build off what she started. But then we're also going to be looking at behaviors, for example, how their bodies are oriented, where they're found, movement, et cetera, and see how the blue lights might be affecting them. So a quick summary of this is really that there's still a lot more to be learned about salmon excluders and salmon behavior in order to get to higher escapement rates and to get to more reliable escapement rates. And just really to highlight that in developing a BRD, it's really important to think about the differences, but understanding that for this particular bycatch problem, it is very challenging given the similarities between the target and the bycatch animal. I also want to mention the importance of industry. So this, a lot of the research around salmon excluders has really been driven by the fishing industry themselves, and it's been a really important and key part of the work and the collaboration. So being able to, on the conservation engineering side, bring in some of the tools that I've highlighted today in order to augment and support the work that's being done with the industry, trying out different designs, and also really the importance of the collaboration with the industry and other scientists being involved in our projects and likewise with theirs. And along those lines, I just want to highlight that Trident Seafoods themselves are taking on some research projects this summer. They're going to be starting soon, and they are working with a boat that has a flapper design, and they're going to be testing the blue lights in that design and placing those in the escapement path to try to make that more perceptible. And they're also going to be trying out using the strobing white lights after the excluder to see if that actually does give the salmon pause and encourage them to move up and take advantage of that area. So they're carrying forward some of this research this summer, and I'm excited to see what they're finding. I also want to mention that, as you probably have realized, that it's really important to think mechanistically about designing these salmon excluders, not just the salmon excluders, but any bycatch reduction device. But for salmon excluders in particular, that location and design of the escapement area is really less limiting than some of these other variables, these other elements that I've highlighted. So it's really important to design these with the behavior of the salmon or the bycatch animal in mind in order to really be more efficient, most efficient and effective in thinking about how these BRDs are designed. I also want to highlight that the innovative tools are really needed to address some of these more challenging problems especially, but they're also becoming increasingly available, and I'm going to talk briefly about some of those. And so as far as the next steps in our group, in the conservation engineering group, we're going to be generating the results from the blue light study. David Bryan is going to be wrapping up the analysis of that, the triggers for the forward movement. And I also wanted to highlight the work by Catherine and Casey Wilson in our group. She is developing artificial intelligence and machine learning platforms in order to detect, do auto detection of salmon in these videos that we're collecting in order to expedite video review. As you might imagine, we have a lot of video, and so through the use of AI, she's able to help us expedite that review. But she's also using this tool as a way to try to estimate the loss of pollock. So looking at the amount of pollock before and after a salmon excluder. So her current, the current model she's got is getting 96% for salmon and pollock detection, and she's continuing to improve those models. Along those lines, she's also working on having these counters programmed in so that you can actually have the ability to count salmon as they go past the camera. So as you might imagine, really exciting things ahead and using some of these new innovative tools in order to address salmon bycatch and bycatch mitigation in general. And just as a final thing, and just to really get folks excited about some of the different work that's being done in order to really think about how to mitigate salmon bycatch. The project, this project here that I'm going to talk about is by Sabrina Garcia, who's at ADF&G, and she's doing her PhD right now at UAF with Dr. Seitz and Jordan Watson and myself. Both at the Alaska Fisheries Science Center are collaborating on this project, which was funded by the Pollock Conservation Cooperative Research Center. And what she's going to be looking at is whether or not we can predict where salmon are going to occur before fishing. So trying to think about where we might have interactions with Chinook salmon for the pollock fishery before even leaving the dock. And her project is going to be looking at whether or not you can determine persistent hotspots or predict persistent hotspots by creating these species distribution models. So for example, there's an example output of Chinook salmon distribution using temperature and chlorophyll data. And in this image here that was provided by Sabrina, the red areas are indicating a high probability of Chinook salmon occurrence. So she's going to be developing these models both using environmental and other data, also informed by collaboration with the fishing industry, and using tracking salmon tagging data that was collected in Dr. Seitz's lab, and then taking that information and overlapping it with the fleet effort and where pollock are caught, and trying to get at whether or not we can use this information to predict likely bycatch occurrence. And as this is her project, I'll provide her email address and direct you to her for questions on that. But really just wanted to give one more piece of the puzzle and these different conservation engineering approaches that we can use to support sustainable fisheries. And I want to acknowledge the conservation engineering group that I work with at Alaska Fisheries Science Center, and again to really highlight the research partners that I've collaborated with from industry and science have really been the key to success for this. And then to also mention that the National Cooperative Research Program and many in-kind contributions is what has made this these projects possible. And with that, I want to thank the Halibut Commission for inviting me and for all of you and your attendance today and getting to talk about conservation engineering and the work that we're doing. So thank you very much for attending and for your time. And with that, Claude, I'll take questions.
All right, thank you very much, Noëlle. It was a very informative and interesting talk. I'll encourage participants to submit questions via the questions tab, and I can read those out when they come in. I had a question, or more of just an idea that maybe you haven't looked at, and it was interesting to think about the concepts about how animals behave, well, how the gear itself behaves and how we assume it behaves, and then how the animals themselves are actually behaving around it. And it got me wondering, with the observation that there's more forward swimming and more escaping happening once the trawl boards are aboard the vessel and the nets coming up towards the surface. Is there another sense that would also help them be motivated, such as a startle behavior that might help them suddenly go into a burst swim pattern and take advantage of their escape location in the form of noise? Is there any evidence that salmon exhibit like avoidance behavior in the presence of killer whale sonar frequency or dolphin whale frequency, where you could have them schooling up towards the surface as you're slowing the net down and bringing it up, and then you would combine that with a sound cue that might also make the strong swimming salmon quickly escape the net?
That's a great question, Claude. Yeah, I think that's something definitely I've thought about. In conservation engineering, I think there's often the temptation to kind of create these like elaborate contraptions and things. So I tend to kind of always work on the KISS principle, keep it simple, silly, and start with like the bare bones easiest thing and then kind of work from there. But it is on my radar to think about that. There is a company out of Austria that also is based in Miami called FT Tech, and they've actually developed these sensors that you can program different noises in. So they've got killer whale, they've got, I think, squid, they've got crab eating, they've got all these different sounds, and they sorry, I warned you, Claude, sorry about that. Had to be 12, the most we could have. Sorry, cuckoo clock. But they do have killer whale sounds in these devices, and it is on my radar to think about maybe using that if we're kind of getting to a place where the lights and things aren't working. But I always try to think about the simplest thing to minimize impact on operations to the fishing crew. So it's a really good idea, Claude. It's on my radar. And again, I don't think there's been a lot of work done in thinking about these noise deterrents, and a lot of the work and thinking about salmon behavior is actually done around dams and sort of obstacles like that. So they're actually in tandem to what we're doing. They're trying to also think about how to affect salmon behavior, and they've done a lot more work with light, and there's been less done with noise, but it's certainly a possibility, and I think it could be we have to get to that place to test that if we don't have as much success with the other things. It's great thought.
Yeah, so I was part of the depredation conference that happened, or whale deterrent conference that happened last week, and one of the things that was discussed there was the startle response and how that's being used to the advantage of keeping sea lions and seals away from gear, where pinnipeds in particular have a very strong flinch response to certain noise triggers, and just it got me thinking about it with fish as well. So
Yeah.
we haven't had any questions through the questions tab. I don't know if people can't find it or you answered all their questions, but we'll keep it open here for another minute or two to see if we get any interest there.
Great. It's hard to believe I answered all the questions. Any other ideas? I'm also open to ideas. It's always great. I feel like giving these seminars, people often suggest things and I hadn't thought about that. So yeah, I'm very open to hearing if other people have ideas as well.
Sure. We're getting just a compliment from Scott Mazzone indicating it was an excellent presentation.
Well, thanks, Scott. Appreciate it.
Well, with that, I would encourage people to reach out to Noëlle. Her email's on the screen there. And I'd like to thank you, Noëlle, for your presentation and for audience members for your time today. A reminder that a recording of this presentation will be made available on our website probably by the end of the week. And you're encouraged to share that with others. And please stay tuned for other installments of IPHC seminars in the future. Thanks for your time.
Thank you for the invitation, Claude.
You're very welcome.